Hey, good afternoon. So my name is Vaughn Tlung,
And my name is Sydney Brown.
and we're presenting our project, Cyanobacteria Community Analysis and Cyanotoxicity Testing in Lake Sinclair, Georgia, guided by Dr. Kleena Malone.
So to give you guys a little bit of background information on cyanobacteria, they're a major player in harmful algal blooms, and they can produce toxins. One of these toxins are microcystins. As you can see on the left is the chemical structure. They have 250 plus structural variant. And so we currently have different methods of detecting cyanotoxins, including satellite imaging, enzyme-based assays, and immunoabsorbent assays. So the World Health Organization and EPA both have guidelines for microcystin levels in drinking water and recreational water. So for drinking water, they suggest less than 1 microgram per liter. And for recreational water, they suggest less than 8 micrograms per liter.
And so looking at the protocol for serving lakes, we mainly focused on the biological and recreational aspects because they had the most to do with algal populations and counts.
So why should we be worried about microcystins? If you look on the left, there's a news article where a West Palm Beach dog had died after being exposed to cyanotoxins. There are different factors which influence toxin production. High temperatures are going to increase algal cell growth and algal density, which has increased risk of cyanotoxin production. There's many different health impacts. Over long term, they can be carcinogenic and they are a hepatotoxin. They're also resistant to destruction.
And so looking at the ways to detect the toxins in the community, we focus mainly on the enzyme-based assay test. And so one pro of this method is that we have quick turnaround, meaning we could do these in the field and within a certain amount of like a short period of time, we can get the results quickly. However, there is one con, which is that it doesn't give us the exact concentration of the microcystins that we want to test for. It only gives us the levels between 1 and 10 nanograms, which isn't feasible for larger aquatic communities or locations.
So our research question was, how can we accurately assess HAB presence? And our hypothesis was that species composition and abundances are a precise methodology for HAB assessment. So the goals of the study were to relate algal biomass with species level identification and abundances. And we achieved them by sampling aquatic communities in Georgia, and we utilized the research grade microscope to detect the genus Microcystis. And then we further identified Microcystis to the species level. We also utilized rapid testing, our test strips, to detect microcystins, and we compared species presence with toxicity levels and chlorophyll A levels of samples. And we analyzed the data to find the best detection methods of microcystins.
Okay, so from the samples that we collected, we found more macroscopic morphological presence of Microcystis aeruginosa, and these are some of the pictures we took from the sampling sites. And on the right here, you can see the picture of Microcystis aeruginosa under the microscope.
So when pairing it with chlorophyll A concentrations, we looked at Lake Sinclair and found that there is a mean chlorophyll A concentration of 1.6 micrograms per liter. And the transparency mean, which is related to chlorophyll A, was 91.11%. And we also found low levels of microcystins in this particular sample, between 1 and 2.5 micrograms per liter. And we found that the chlorophyll A was significantly different, as well as the transparency.
And so here we have Cylindrospermopsis raciborski. So we found these in Lake Sinclair as well. And looking at the cell on the bottom half, you can see the the presence of heterocytes, which is indicative of this species.
So going into Microcystis species, Microcystis aeruginosa can be identified morphologically through the formation of clathrate colonies, as you can see on the left. And Microcystis botrys forms clustered colonies which are not connected. And Microcystis wessenbergi forms colonies which are bound by a mucilage sheath, as you can see better on the right. And for our test strips, as you can see, quick and easy method, A and B show replicates from Lake Sinclair, and you can see that the control line on the left is darker than our test line, which is indicative of microcystins.
And so we ran EPA toxicity tests for 4 outwell toxins, and we confirmed that there were cylindrospermopsin and microcystins produced by cylindrospermopsis and microcystis in concentrations lower than 4 micrograms per liter in the drinking water. Anatoxins and saxitoxins were not detected, and community biomass measured as chlorophyll A were never above 5 micrograms per liter. But we also found that, most importantly, algal communities were dominated by diatoms, and we did species level identification of toxigenic species such as Microcystis aeruginosa.
So the EPA has developed this tool which predicts the likelihood that we're going to see microcystins in a water column. And as you can see, higher chlorophyll A concentration is a higher likelihood of microcystins present.
And so we were able to do species identification and abundances. We were able to determine that species identification and abundances are more informative and compared to biomass estimates in terms of chlorophyll A. And there's more research investigating ecological drivers of microcystin accumulation, and like they need to be done earlier to detect it before toxic chemicals can be accumulated. And all microcystin colony found in this study were small and non-dominant community, but we were able to find enough to have species level confirmation.
So thank you, Georgia College and State University, the Department of Biological and Environmental Sciences, and the Manoila Phycology Lab for providing the resources and help for this research. Additionally, thank you to the Manoila Phycology class for the assistance in collecting samples and running replicates of toxicity tests.
Here are our sources.